Assalamualaikum and hello everyone. Today we are going to explain the role of biopreservation in improving food safety and storage. First, introduction. Let's start. Contamination and spoiling by pathogens is a great concern by food industry. Food industry make chemical preservative and physical treatment. Also in biotechnology, in food processing, improve the use of non-pathogenic microorganisms. Here are the several bacterial pathogens that can affect human life, such as Salmonella, Campylobacter jejuni, Escherichia coli, Listeria monocytogenes, Staphylococcus aureus, and Clostridium botulinum. Do you know what are biopreservation? Biopreservation is a standard storage life and enhanced safety food using the natural or controlled microbiota and their antimicrobial compounds. Just like that, the beneficial or fermentation product used in biopreservation to control spoilage and render pathogen inactive. For example, like pickles and jams. Next, compounds such as organic acid, bacteriocide, triacetyl, and acetaldehyde, also enzyme, carbon dioxide, hydrogen peroxide are used for antimicrobial activity in biopreservation. So we know the need of biopreservation in food industry.
Moving to the next part, let's talk about what is bacteriocins. So, bacteriocins are peptides, or we call as a complex protein, that are produced by bacteria in which it is biologically active with antimicrobial action that against other bacteria and closely related species. Bacteriocins are differ from most therapeutic antibiotics that have potential to elicit allergic reaction in humans and other medical problems. Hence, bacteriocins are normally not termed as antibiotics to avoid confusion with therapeutic antibiotics. Other facts about bacteriocin: it is a proteinaceous agent that are quickly digested by protease in human digestive tract. Bacteriocins are ribosomally synthesized that have possibility to improve their characteristics to enhance their intensity and spectra of action. Production of bacteriocins give benefits for food and feed producers. This is because with sufficient amount of bacteriocins, these peptides can kill and inhibit pathogenic bacteria that compete for the same ecological niche and nutrients. This can be supported by the fact that most of the bacteriocins have a narrow host range and likely to be most effective against related bacteria with nutritive demands.
Now, let's move to LAB bacteriocins, that is stands for lactic acid bacteria bacteriocins. There are a study shows that in several LAB, it can produce multiple bacteriocins in about 2 until 3 bacteriocins compared to other bacteriocins producers that only can synthesize 1 bacteriocin. The production of some class 2 bacteriocins regulated through a signals transduction system that consists of 3 components, which are induction factor, protein kinase, and response regulator. The facts about LAB bacteriocins that we have to know is it has a suitable characteristics that make them can be used in food preservative. The characteristics are They are protein nature, inactivation by proteolytic enzymes of gastrointestinal tract. They are also non-toxic to laboratory animals and non-immunogenic. Next, they are inactive against eukaryotic cells. Besides, they are thermoresistant that can maintain antimicrobial activity after pasteurization and sterilization. The last one is they have broad bactericidal activity that affect gram-positive bacteria and damage gram-negative bacteria.
Now we look on field of application. Before bacteriocin can be applied in foods, their cytolytic ability should be assessed in detail. Recombinant DNA technology is currently applied to enhance production, to transfer bacteriocin genes to other species, and for mutation and selection of bacteriocins variants with increased broad activity spectra. Continuous study of the physical and chemical properties, mode of action, and structure-function relationship of bacteriocin is necessary if their potential in food preservation is to be exploited. Classification. Bacteriocin can be classified into 4 classes. For class 1, it is termed lantibiotic. It is a group of small peptides that are characterized by their content of several unusual amino acids. For class 2, it is unmodified peptides. These are small, non-modified heat-stable peptides. In general, they have an amphiphilic helical structure. For class 3, it is formally termed bacteriolysine, large, heat-level protein with modest prospect as food biopreservative. And lastly is class 4, which is circular peptides. It is characterized by a peptide bond between the C and N terminus. Factors inhibiting bacteriocin production include inadequate physical condition and chemical composition of food, which are pH, temperature, and nutrients, spontaneous loss in production capacity, antagonism effect of other microorganisms in foods, and inactivation by phage of the producing strain. The effectiveness of bacteriocin activity in food is negatively affected by, first, resistant development of pathogen to the bacteriocin. Secondly, inadequate environmental condition for the biological activity. Third, higher retention of the bacteriocin molecule by food system components. And lastly, inactivation by other additives.
In this part, I will explain about the bacteriocins of various gram-positive bacteria. Many studies has been focused on antimicrobial agents that produced by lactic acid bacteria, where lactic acid bacteria is known as a group of a gram-positive bacteria. They found that bacteriocin produced for a variety of gram-positive bacteria such as Staphylococcus, Clostridium, and Bacillus species can control the growth of unwanted microorganisms, especially those that can cause health concerns such as Clostridium botulinum and Listeria monocytogenes. Even if the use of this bacteriocin produced might be a pathogenic, the recent development technique in genetic engineering have managed to make a transfer of gene encoding for bacteriocins production from both gram-positive and gram-negative bacteria to improve food safety and quality. Next, we move to Bacillus as biopreservative. Studies related to bacteriocins production in Bacillus species has been done over the past few decades, which include species such as Bacillus subtilis that produce bacteriocins called substilin, Bacillus megatherium that produce bacteriocin called megacin, and Bacillus stearothermophilus that produce bacteriocin called thermacin. Some of the reasons why bacteriocins produced by Bacillus species could be alternative to those produced by lactic acid bacteria are: firstly, Bacillus species has been used in food making for a long period of time, and various enzymes obtained from Bacillus has been widely used in food processing. This shows that bacteriocins from these microorganisms are safe for humans to consume because there is no unfavorable effects that have been shown from humans that consume foods of products made from these bacteria. Next reason is because Bacillus species has a greater antimicrobial than lactic acid bacteria and their bacteriocins. This is due to antimicrobial action in Bacillus species against gram-positive and gram-negative bacteria as well as fungi. The third reason is the bacteriocins produced in Bacillus species could result various effects such as inhibitory activity at acidic or alkaline condition or after thermal processing. This is because of the metabolic diversity in this species that make it suitable for food processing. Lastly, it is because of the genetics of Bacillus species are well understood, as it has been studied for a long time. So, the molecular biology techniques would provide a reliable and safe tool for producing bacteriocins for the use of food industry.
As we all know, bacteriocins acts as a popular natural preservatives in food industry. However, do you know how bacteriocins are used for preservation of foods? Let me show you. The first one is by inoculation of food with lactic acid bacteria that can produce bacteriocin to the food. This is because the ability of the lactic acid bacteria to produce bacteriocin in the food is important for the preservation to occur. The second one is by adding purified or semi-purified bacteriocins to the food. And lastly, by using a product that is previously fermented by bacteriocins producing strain. A few examples of biopreservation in seafoods. Sarkar can inhibit the Listeria monocytogenes in cold smoked salmon. Not just bacteriocins, non-bacteriocin producing strain of Carnobacterium piscicola can inhibit the Listeria monocytogenes in a vacuum-packed cold smoked salmon. It shows that the non-bacteriocin producing strain of Carnobacterium piscicola is as effective as the bacteriocin producing strain 1. The inhibition of Listeria monocytogenes is caused by competitive growth of Carnobacterium piscicola and leads to nutrient depletion. It is also proven that bacteriocin niacin Z, carnosine U149, and the preparation of crude bavaricin A can extend the shelf life of brine shrimp. For rainbow trout, the inhibition of Listeria monocytogenes and Mesophilic arabidopteria can be shown by niacin, sodium lactate, or their combination in vacuum-packed cold smoked rainbow trout. In conclusion, by using bacteria and also their antibacterial product, biopreservation can extend the storage life and increase the food safety of the foods. In order to inhibit microbial growth, it is commonly used in combination with other conservation factors, which is called hurdles. Not only does it benefit economically, the microbial stability, sensory and nutritional qualities of a food will be improved.